my absolute pleasure to introduce my friend and new colleague, Dionysios Raitsos, to tell us more about large-scale, long-term interactions between the land, the sea, the atmosphere, thinking about ecosystem health. So Dio received his PhD from Plymouth University and then served as a senior research scientist at Plymouth Marine Laboratory from 2013 to 2019. Before beginning at the University of Athens, where he is an associate professor of biological oceanography. And we met up because Georgetown Earth Commons is building a relationship with the University of Athens. And we had an amazing class this summer that maybe you can tell them more about.
Absolutely, I have a slide.
Wonderful, fantastic. And we are going to be working on a jellyfish from space project. So I think that's a new sentence no one's ever said before.
Indeed.
And it's wonderful to have you here. So
thank you very
I will
much
scoot off to the side. Thank you so much.
Good afternoon, everybody. My name is Dionysios, and you're going to bear my accent for the next 46 minutes. So I'm going to talk to you about something that I truly love, the ocean color. It's something that many of us have keep watching, and the differences between the colors in the Mediterranean Sea, in the Atlantic, in the Pacific Ocean, in coastal zones, in estuaries, and we always see a big difference, but perhaps we do not truly understand why we see those changes. So I would like to start with the fact that we live on the blue planet. 71% of our planet is composed of water. The marine environment occupies the vast majority of it. In order to understand the functioning of marine ecosystems, and in order to predict any impact of the pressures from the anthropogenic or not, any variability, in order to predict that, we need long-term data sets. Let's start with the history of ocean color. As you can see, these are like different water masses in the open ocean or in front of an estuary, and you can tell that their color is significantly different. Back then, more than 150 years ago, they wanted to get some oceanographic instruments or lake instruments for limnology in order to capture this differentiation. One of it, it's called SECI, the SECI disk, I think you all know. And for the online people, I'm showing some very interesting stuff. And the foral hull. So this little thing, you just down until you cannot see the white disk anymore. It doesn't sound very scientific, and I have like one even more, the foral hull, the measurements that you can actually see through the hole, and you can tell what's the color of my jacket.
Green, blue.
Now look at the little numbers.
Oh, the number, sorry, 3.
Number 3, so pretty eutrophic. I agree with you. So it doesn't sound very scientific. I mean, yesterday we went, not yesterday, a few days ago, to the Potomac River with a few students to sample the waters. I got a very interesting question. The vast majority said number 8, 9, and students, I don't know the percentage, they said like probably 14, 15. And then I got the question, the fact that we have like one measurement of foral hull and we know the scale from 1 until 21, is it important? The answer is no. However, if we get hundreds of those, because it's a very simple equipment, does it truly work if we have plenty? And here I'm presenting you the first map, global map, and this is only the Atlantic, but there are other seas as well. The first map of primary productivity in the open oceans, but it's nearly 100 years old. So what did they manage to see? That in the open ocean, the shaky disk, but also the color of the ocean is mainly deriving from biological processes. So we're talking about phytoplankton. Phytoplankton that they have such an abundance that they're capable of changing the color of the ocean. This, in order to realize what did they manage to do, important to mention, since not only the scientists were using, but any sailors, anybody that was traveling from the UK to US, vice versa, from Africa to Europe, in whichever ships, scientists were giving those shaky disks, which were very easy, and they were asking the sailors to take a note. By doing that for decades, they created this map. So in a sense, I believe we can call them the first citizen science projects. Let's see how do they relate with reality from satellite-derived information. Look at the oligotrophic gyres, the anticyclonic gyres in the north and the southern hemisphere, which are the biological deserts, exactly at the same position. Look at the upwelling system in Africa that leads up to South America, and we can see that here. Have a look at the southern Latin America, that we have very eutrophic waters. Exactly the same thing in the northern Atlantic. So it is actually picking up every single detail. And have a look at the Mediterranean Sea, one of the most oligotrophic seas in the world. However, the eastern part of the Mediterranean, it's the most oligotrophic. Look that the Forelule measurements were capable of separating the ultra oligotrophic waters from the oligotrophic waters. So we're talking about an astonishing oceanographic equipment that was developed 150 years ago with shaking measurements that they would measure clarity, and later on with Forelule and what we can actually get. One of the pioneers of ocean color was Sir Alistair Hardy from the UK. What he did, first of all, he discovered and he designed the continuous plankton recorder, which it is the longest biological data set in the world. They have collected more than 600,000 samples all over the globe from the continuous plankton recorder, zooplankton, phytoplankton to species level, more than 1,000 taxa and species identified. His life was plankton, as you can imagine. It happened to take a plane and fly from Plymouth over Cornwall and differentiate the green water, the blue, and the brown. 100 years ago, in his book, he said, if these marked color changes can be correctly interpreted, we may in the future find aircraft being used to make rapid surveys of surface conditions in relation to fisheries. So back then, scientists realized the importance of the ocean color, and they started linking already from back then to commercially important industries. NASA was the first entity that created and launched a satellite to monitor the marine environment called CZCS, by having sensors that they measure the light that is absorbed, reflected, and scattered, and like that, learning about characteristics of the ocean surface and estimate the concentration and various types of matter suspended in seawater, including phytoplankton, sedum, sediments if it is in a very coastal environment. So basically here in the video, as the Earth rotates, the seasons are changing. And as they change, you can see the North Atlantic that is blooming. On the land, you can actually see the chlorophyll biomass, the MBVI, and in the ocean, also you can see the ice coverage. Look at that during the winter time. But when it starts melting, we're going to start seeing blooms, the spring bloom of the North Atlantic. And by observing the Earth every single day, the last 30 years in terms of ice, in terms of plant biomass on land, but also biomass in the oceans, we start having a huge quantity of data sets. And the accuracy, it is impressive. When I started my PhD, only sea waves was alive, and it was just the beginning of MODIS from NASA again. Since then, that's why I'm having this image from the European Space Agency. They started with MERIS and monitoring the environment 20 years ago, and we can see how many satellites have been launched to monitor the environment in exceptional resolution. So NASA and ESA are working together constantly in order to have, for instance, NASA has PACE at the moment, which is a hyperspectral sensors on board, astonishing quality, and we're looking forward to play with the data that was like launched very, very recently. Why does NASA care? Why do ocean color scientists care so much about measuring the ocean color? Because in the open ocean, it mainly comes from phytoplankton. There is no little centimeter at the surface of the ocean without having these microorganisms. They live in every aquatic environment. They're an integral part of the ecosystem. And they are capable of absorbing for the photosynthesis. They take carbon dioxide, they remove the carbon, and they repel oxygen. So they oxygenate the oceans. However, not only the oceans, 50% of the oxygen that we breathe in this room is coming from phytoplankton. So basically, every second breath you take, you owe to phytoplankton. They're growing in enormous quantities. They're capable of changing the climate. And climate, especially change, unfortunately changes phytoplankton back. How do they look like? Diatoms, dinoflagellates, silicoflagellates, coccolithophores. Very beautiful to see them under the microscope. This is, for instance, from the Potomac River, a very nice diatom chain. They aggregate all together. They create a huge chain. They look huge, although they're very tiny. And like that, they're repelling the potential predators. So the question is, what limits phytoplankton growth? There are two limiting factors. Of course, salinity, temperature, current, waves, everything is impacting the ecosystem and phytoplankton. And these are indirect measurements in order to be able to see the variability and understand the variability of phytoplankton. However, there are two limited parameters, the light and the nutrients. The light, as you can see from the satellite remote sensing, here, it's pretty standard. In the tropics, we have a lot of light. In the temperate seas, a little bit less. And near the pole, we have minimum amount of light. That's why Donne et al. In 2006 in Nature, he split the ecosystem, the marine ecosystems in two. The higher latitudes, which are light limited, and the lower latitudes that are nutrient limited. Why they're nutrient limited in the tropics and the warm temperate seas? We're going to discuss it now. So in order to understand the phytoplankton environments, we need to understand where do the nutrients come from. They can come from the atmosphere through aerosols, dust, black carbon, but they can come from rivers, huge quantities of nutrients. They can come horizontally, but one of the key mechanisms is the vertical mixing. Let's talk about the norm. The norm, during the winter time, we have a lot of mixing. Why is that? Everything is related to physics. Although I don't like physics at all, and physics doesn't like me either. I have taken several courses in physics. However, unfortunately, the biology and the whole ecosystem depends on physics, on density. So basically, during the winter time, the air temperature is cooling down. By cooling down, the surface of the ocean starts cooling down. The moment the water cools down, it becomes more heavy. The density is increasing. Whereas when it is warming up, it becomes more light. So during the winter time, this surface colder, denser water is sinking. Of course, it doesn't leave a hole at the surface of the ocean. It is sinking and replaced. Again, the next layer is again cooling down, sinking, cooling down, sinking. And this is the process from the autumn until the winter time, that there is homogeneity. As you can see, this dotted line here, it's the temperature. The temperature at surface and at 300 meters depth is exactly the same. Whereas during summer, we can see it is way warmer, then there is an abrupt change. This abrupt change, it's at the level of the thermocline. Why is that? Because basically, the densities of the densities of the water masses are so different in terms of temperature driven or salinity driven, the more salt, the more heavy. So either if it's temperature or salinity driven, the two water masses cannot mix. At the same, exactly at the point that they mix, it's called thermocline. It's like having in a glass oil and water. They will separate because they have different density. Why do we care about that? Because this is the line that is holding the nutrients below the thermocline and does not allow the nutrients to come up for photosynthesis. As I told you, it's a limiting factor. So the nutrients are trapped. Why the nutrients are down there? Because everything that lives on Earth will become nutrients like us. So this is the positive perspective of the day. And when the water column is mixed, we can see that immediately we have a very high abundance of phytoplankton. In red, it's the temperature, February and March, the peak of winter. And here we can see the peak of chlorophyll, whereas when the summer starts and the summer stratification starts, it is immediately nearly finishing and not having any very low concentration. So it's totally anti-correlated. What's the problem? The problem is with the climate warming, because what I explained until now, it is a pure seasonal cycle. This is derived from 25 years of satellite derived information. It's great, but this is the normality. During winter, we have a peak. During summer, a low in chlorophyll, driven due to the stratification. The problem is, what is happening when we have a much warmer winter in comparison to the normal? As we all know, climate change is changing, truly changes the Earth's surface, the marine, the terrestrial ecosystem. It's constantly increasing, and we are responsible of that. You all know, I don't need to say anything more. Let's take an example of the Mediterranean Sea. This is like time series anomalies, basically the deviation from the average, from the mean, from the overall mean. Whatever goes above, it's higher temperature. Whatever is below this line, it's lower temperature. This can be also plotted with colors, so from 1985 until present, and from January until December. This is the Eastern Mediterranean Sea, and this is the temperature of the Mediterranean Sea. Next to it, it is the temperature of the whole Northern Hemisphere. As you can see, they have changed evidently from 1998 onwards, one of the biggest El Niños we have ever had. The whole Earth was warmer. And on this figure from 1950s until present, we can see that we are talking in the box during this period of time. So we live in a very interesting era of getting a lot warmer. What is happening with phytoplankton? Let's compare. Let's avoid the seasonal cycle and let's compare a cold winter with a warm winter. And I'm going to take you in an oasis of the Eastern Mediterranean, which is the only blooming compartment of the Eastern Mediterranean. It's extremely oligotrophic, but it has Rhodes gyre. And this is a cyclonic eddy that is constantly moving and is bringing nutrients up. It is a permanent feature, however, during the peak of winter, wind-driven forcing and also the vertical mixing from cooling, it rotates the gyre even faster and it brings all the nutrients up, creating huge blobs. Look what is happening during a cold winter and look what is happening during warm winter. Nearly, and in this particular winter, we had a marine heat wave, nearly disappeared. In order to visualize that better, using a BGC Argo float, a float that takes measurements at surface, a whole water column, one reaches the bottom, it goes up again, and transfer all the in situ data sets back to the lab. And it can survive for 5 years and provide amazing data set to the community. What we can see, this is the stratification. So it's a measure how well the oceans are mixed. Have a look at the winter. At the peak of the phytoplankton, the temperature from 0 until 250 meters depth, it's all homogeneous. It is fully oxygenated and chlorophyll is in the whole water column. Is indicative of the phytoplankton biomass. Exactly the opposite is happening during summer with thermal stratification and minimum amount of chlorophyll at surface. Going back to the Mediterranean, you can see what is happening during a marine heat wave, which is the extreme version of warming. In addition to the long-term warming, we have some extreme events called marine heat waves. First introduced by Hoptey et al. In 2016. And we have the marine cold spells. Have a look at the difference in the quantities of phytoplankton biomass in comparison to those two cases. This translates, as you can see on this, it truly translates into fisheries. And look at the landings during a very productive year and look at the landings during a non-productive year. So remember, mixing enriches, stratification inhibits the nutrients. And in green, it's the chlorophyll, the satellite-derived chlorophyll. And in black, not black, in orange, it's the mixed layer depth. So it is the depth of the mixed layer. Look how tight this relationship is. So basically, the deeper the mixed layer, the higher phytoplankton biomass. So this is over Canary Island. So we're talking about these relationships are changing due to warming because the warmer the water is, the less mixed layer depth we have. So basically, the less mixing. The question is, could such relationship affect higher trophic levels? No. Of course, yes, otherwise there wouldn't have been. So let me show you an example in Tanzania, in Africa, that we work with colleagues from there and with Southampton, a very nice study that we did. In purple pink, it's the landings of small pelagic fish like mackerel, horse mackerel, anchovies, herrings, and salts. And in green is the satellite-derived chlorophyll. You can see how much fisheries is related to the phytoplankton variability. Any change in the phytoplankton biomass, as we can see here, it is truly have an impact on higher trophic levels. So it is quite important. And we can see that this variability in chlorophyll, in phytoplankton biomass, it's purely driven by environmental factors like temperature, for instance. Another example, in South Africa, with choka squid, one of the most important squid fisheries in the region. I think the most important. We're very lucky through a nice program called Solstice, led by Southampton, that we were having access to some great data sets from the ministry, monthly choka squid fisheries landings all over South Africa. And this is the relationship that we get with chlorophyll. And in the papers, of course, we have the full mechanism, because the mechanism, it's not simplistic at all. And you can see how climate warming is impacting the amount of nutrients, and eventually that escalates to higher trophic levels. Warming affects not only the abundance of phytoplankton, but also the bloom timing, the well-known phenology. Like in terrestrial environments that we have, like the larvae, and the transition form of an organism during its development from the embryo to a mature individual, and then it becomes a butterfly, and this stage is called metamorphosis. The interesting part is that in the marine environment, many species, they have these metamorphosis stage. So actually, after the egg, even if you are a lobster that lives in 2,000 meters depth, or a fish that lives in 3,000 meters depth, you have a pelagic state that after the egg that you hatch, you go up. How do they know where to go when you are at 3,000 meters depth? Basically, phototactic. You're following the light, the sun. Even if you cannot see anything, a little bit, you can have the direction. That's why if we even want to collect zooplankton from an already good sample from a tank, or the larvae, or ichthyoplankton, if we have any spawning, even from the aquarium, we just turn off the lights, we take our mobile, turn on the lights, put it next to it, it aggregates all the larvae, and you collect easily. So imagine, it's called metamorphosis because this doesn't truly look like lobster, that doesn't truly look like the gilded sea bream that we see here. Larvae and their ecological traits play a very important role for the marine communities. And here from my lab, that we have, we are running experiments on Palaemon elegans, the coastal shrimp, and look at the larvae. How does it look like? And eventually, it will get transformed to an adult. It doesn't matter if you're a crustacean, if you're like a fish, and many other organisms, you have these planktonic states at the beginning of your life that's feeding upon the gold from through photosynthesis that we have. And this is like a clownfish in the lab that we have. The moment that they hatch, we have to feed them with phytoplankton and rotifers, very slow zooplankton, because they can hardly reach it. So the copepods are for later stages, imagine. So they need something that is very, very slow. So the phenology, the timing of phytoplankton relates to the, it's a biological event, typically primary producers, and we have it on land during the spring, blossom, and exactly the same thing is happening. Look at the seasonal cycle of the North Atlantic Ocean. It is peaking during spring, and then and we can see this very standard seasonal cycle. And there are many metrics that we can measure the phenology. We have the initiation, the duration, the peak, and the termination. Why does that matter? This is called the match-mismatch hypothesis, published by Cushing in 1974. It is quite interesting. Have a look at the blue part, which is the larvae. The larvae in the organism start reproducing before the food arrives, because when the food arrives, it's already too late to start reproducing for the larvae to be ready to feed upon phytoplankton. So they reproduce and they're expecting the food to come. The food is in yellow. Imagine, and this is time. If phytoplankton bloom delays, the amount of time that larvae have on phytoplankton reduces. They call it a mismatch. However, if it comes earlier, for a few weeks, it can be even more beneficial than the normal case. If it comes 6 months earlier, again, it's going to be a mismatch. So timing matters significantly. There are some very nice papers by Trevor Platt in Nature showing the impact on haddock and with the crustaceans again in Science. Very, very nice papers. The zooplankton includes an exceptionally high diversity of larvae and adults for most animal phyla and many protozoa. To take a break, I'm going to show you some live specimens. Yeah. Through a microscope. Right. Go back. Right. That's positive. Right. We'll move. Let's fix the light. All right. As you can see, those two are the eggs. I collected those. We went for a romantic trip in New York with my wife, and she thought that we indeed going to have a romantic holidays. Of course, the microscope was with me, along with the plankton net. So we were seeing the sunset, and that is the best time to collect zooplankton, actually, in the phytoplankton. So I was particularly happy. And then I transferred from New York here, and it was like pretty. Poppy pods, one of the most nutritious, and I think I have a third one somewhere here. One of the beauties with this microscope, the Curiosity microscope, that was only the first. They were kept alive from Long Island, these New Yorkers. Oh, it's moving. Hello. And this is Zoom at 50%. It's a very, very nice educational tool. It doesn't have any drivers. You just, it comes along with the plankton net. So everything has been collected. It's 20 microns. Pretty fantastic equipment. It has everything inside. It connects, doesn't need any power. So it's a very nice tool. I'm always having it with me. So these copepods were like kept alive in a jar with 1 liter. All the phytoplankton died. I haven't brought all the equipment to keep it alive, but this zooplankton 20 days ago did not have any eggs, so they have been in business. All right. So the phenology, how do we calculate? There is an algorithm derived by Trevor Platt and Subhash Andriandrana, back in the 90s, and it's further developed by the international community. And there is a very nice, using the cumulative sum of anomaly, and you derive the initiation, the peak, the duration, and the termination. And you can apply that in every single pixel all over the globe. And you can identify when does it start, how long does it last, when is the peak, when does it finish. Is it accurate? Pretty accurate, using 25 years of in situ data set over a coastal station in outside Athens. We can see how it relates with in situ data, but also following an Argo float, you can see how much they're related and identified nearly overlapping exactly at the same period of time. Having this information, what you can do, for instance, I'm showing you an example from my PhD student, Antonia Kournopoulou, and you can see, let's go, for instance, here in the purple. What you can see is that the peak of the phytoplankton, it is taking place in May. And then you have all the different metrics. So for every pixel, you know exactly. So it is quite important because it can derive information that it can be useful from every marine ecosystem. There are like some global works on that by Marie-Fanny Lagracolt, excellent papers if you're interested in phenology. Trevor Platt started his Nature paper in 2003, and in the abstract he mentioned the link, this link between phytoplankton phenology and fish larvae has been suspected for more than 100 years. What else would they eat? But its verification had to wait for technology with sufficient spatial and temporal resolution. Indeed, he went out in the middle of the Atlantic collecting haddock, a species that live like in 300-400 meters depth. Look what he finds. This is the survival of the fish larvae. Whenever the spring bloom of the Atlantic was 1, 2, or 3 weeks earlier, the survival rate has increased massively in comparison to the years that it was 1 or 2 weeks later. So basically, he proved that when the phytoplankton bloom is moving, you have less quantity of food. But for a species that lives in 300 meters depth, which is pretty amazing, whereas the phytoplankton lives at surface. In a study that we try to see if these mechanisms are evident in the Red Sea, here you can see that the deeper the mixed layer, the earlier the bloom. Whereas when it's warmer and the mixed layer depth is shallower, the bloom delays and it's later by 4 to 5 weeks. When we saw that, this differentiation, we wanted to add some fisheries data in order to see what is happening and how the fish are related to those changes. And I'm showing you here, I'm showing you here the seasonal cycles of phytoplankton biomass in gray and a very peculiar year in blue. In 2003, we had the largest bloom of phytoplankton in the sea. During the winter time, it remained like at normal, not low, not high, very normal. However, the bloom started many months before. So when we saw that, we suspected that for fisheries, the year after, because they need to grow in order to be caught, and we targeted the small pelagic and the small squids, we wanted to see if there is any impact. We calculated the phenology. So basically, in 2003, you had the initiation approximately 6 years, sorry, the initiation 6 weeks earlier. The termination was approximately 4 weeks later, and the overall duration approximately 10 weeks longer. And this is the result. On the left-hand side in green, you have the phytoplankton biomass, and in black, you have the Sardinella species. and below you have the squid species. And you can see how they're related with a year lag, and how much reaching like 1,000 of metric tons, whereas the usual one was approximately 200-300. Warming influences fish not only indirectly through the food availability, but also directly by affecting their immune system. And I'm going to show you a very nice study that we did with colleagues in the Gulf of Aqaba. You can see that the lionfish is not swimming very well, obviously. And you can see that on the left corner, you have groupers, snappers, parrotfish, pufferfish, almost any size, from 10 kilos, 15 kilos of fish, until a very small one, from even damsels. They were all swimming like that. So people went out, scientists, they collect them alive. It is very important to collect alive, because if you have a dead fish in your fridge for a week, of course, it's going to be full of bacteria. What we found is that there was an abrupt shift, a steep rise in water temperature of 4.2 degrees of Celsius within 2 days and a half. This is nearly impossible. This is nearly impossible due to the thermal capacity of the sea. We didn't believe. So it was very good that we had in situ data sets, as you can see, the light blue from the Pierre, 3 measurements per day the last 20 years, and the satellite derived, which they said exactly the same thing. And this is the relationship, it's super high, and we have further statistics. What happened? And this mechanism, until now, it was proven only with experiments on animals. They turning on the heat of the aquarium, increasing by 3-4 degrees of Celsius, and they were like spotting the bacteria infection. 95 of the live fish that they were caught while swimming like that, they have encephalitis, and they were infected by the bacterium Streptococcus pneumoniae. What happened? If you have an aquarium at home, you can realize that the fish can get very easily stressed. Lack of oxygen, or increase of temperature, or increase of salinity, decrease of salinity, whatever parameter changes rapidly, it is a huge stress. The fish, like human beings, the moment we are stressed, our immune system is dropping down. Adrenaline is keeping up, but especially when the stressor is moving, this is when the immune system reaches the bottom. So basically, we had a huge mass mortality due to temperature, and this is like a very direct example. What happened? We have the full mechanism, but very briefly, thousands of kilometers away in the Sahara, there was like a thermal low so strong during July, and these very warm atmospheric masses were moved in the surrounding, in the Mediterranean, in Africa, and hit the northern tip of the Red Sea. And it created, only for 3-4 days, this particular wave, and after a week, it happened again. It's amazing that in between, there was no signal at all above Africa. So you can understand that changes in the terrestrial environment have a huge impact on the marine environment. Another way that warming can actually impact the marine ecosystem is by the introduction of new alien species, or otherwise called non-indigenous species. So this is a very long time series, nearly 100 years old, linking the new entrance of alien species with temperatures over the Mediterranean Sea. In the Mediterranean Sea, we have like more than 1,000 species. And this link, the species that I used here were only the warm tropical species. We can discuss later on how did they end up in the Mediterranean Sea. it is not only affecting fisheries, it is affecting aquaculture. So it has like a huge impact in aquaculture industry. We published recently a review of the mussels and bivalves aquaculture and the impact of warming. If it gets too warm during marine heat waves, for instance, in 2021, we can see that the bivalves are suffering from pathogenic microbes. We want it to continue, and I'm feeding my clownfish in order to reproduce with live muscle. If I don't give them live muscle, they don't reproduce. They're very elegant. And so in last September 2024, I went to the fish markets, I couldn't find. I sent all the students all around Athens, nobody had. After a month, of course, our clownfish stopped breeding. We were very disappointed about that because we had a long ongoing experiment that we wanted to run. Basically, we didn't find any mussels, and around Christmas, we started finding Spanish mussels. Basically, there were no Greek mussels in the entire Greece. So I started calling the directors of big fisheries landing places, the fish markets, and one of them was particularly helpful. He told me that after the marine heat wave that we have in July and August, nearly all the aquacultures have been destroyed in the northern Aegean Sea, exactly where 90% of the yield is coming. So the 90% of Greece supply comes from the northern Aegean Sea. It's pretty eutrophic environment, a lot of phytoplankton, many rivers, loads of nutrients. And what's the problem with climate change and oceanic warming and aquaculture of mussels? Is that Mytilus galloprovincialis, of course, has got used to the warm climate of Greece. However, around 26 degrees, it stops feeding. 27 degrees, absolutely, the filtration doesn't want to continue. So basically, it starts shrinking. If the warming it's more prolonged, like the duration of the marine heat wave, that's why it's extremely important to measure it, then they literally stop feeding and they're becoming very small until the level, if it is prolonged enough, they die. Have a look at more than 20 years of time series of monthly aquaculture yield. Look what happened in 2024. So we're talking about the aquaculture collapse. Even the government at the moment that we're talking doesn't know how to control it and what to do. Indeed, the 2024 was an exceptional year. And on this, quite interesting to say that it's not only in 2024 that we see the impact, but the two time series, they mirror each other in a negative way, which means any variation, if it doesn't cause a big damage, any variation in temperature during summer, it is impacting the yield. This figure, it's the number of marine heat waves, the number of events that has happened in comparison with the mussels. As you can see, no heat waves before 1998, absence. The first marine heat wave, pretty small. And look at the exponential growth of the marine heat waves and the cumulative intensity. And because we don't care about the environment, but we care about the finances, look at the sales, look at the way that they're shrinking. The sales and the value is dropping massively because the cell remains the same, but when you open it, it's empty. Nobody wants to pay for an empty mussel. There is only one ecosystem, and this is called Earth. And I'm going to finish the talk with two examples. We all remember in 2019, the fires in Australia, devastating fires that traveled the media of every part of the world. It's very weird that we can have like forest wildfires, desertification of lands, and Oceanic Productivity as a title. It's a fantastic study that they did from Plymouth Marine Lab, from Princeton University, many colleagues all around the globe, and they published in Nature a very interesting study. So basically, in the Southern Pacific Ocean, there was like a huge bloom that occurred in 2019 towards the winter of the summer of the Austrian summer of 2020. After the fires, exactly after the fires, you can see all the smoke coming out and the black carbon. By plotting the Aeolian trajectories, you would follow where the winds will go. So what happened? The black carbon, which was at its peak, carrying nutrients like iron, it went to an iron-limited area, and it's just rained, nothing more, nothing less. So we're talking about wet deposition of the whole atmospheric column onto the sea water. We start seeing more and more often examples like that. We visited a typical blooming place, the Madagascar, huge blooms. Usually it's a winter blooming environment, but it has these summer blooms that they get quite often, every 3-4 years. There was an astonishing bloom exactly at the same time that the Australian fires happened. Unrelated, however, we are talking about one of the warmest years on Earth ever recorded. Look at the phenology. These are only the blooms, the big blooms, the very rare big blooms, approximately 7-8 big blooms. Look in green, the bloom of 2019-2020. It happened 3-4 months earlier. It started in November, and it's 250-300% higher. The air temperature is increasing. The drought index shows clearly that after 2012 is decreasing exactly at the same time with air temperature. The soil moisture is decreasing. All these warmer temperatures that we are going through, of course, they dehydrate the soil. When you dehydrate the soil and you have lack of moisture, it becomes dust easily to move out. This is Aeolian, the dust plotted, satellite-derived information of dust and beautiful models that they model the dust. And you can see the Aeolian trajectories. Using backward modeling, you can ask from where did the wind and the dust came from. So it led to this particular box that we have taken this time series and showing what has happened in the period, causing one of the largest blooms. So basically, I'm going to let the tripod dinoflagellate tell you goodbye, but I want you to focus on the flagella. Dinoflagellate, it comes from the Greek word dinos, which means whirling or spinning. The flagellum, it's the whip-like appendages that it has. So this very beautiful image, we were so excited when it happened that we start shouting because we literally saw the flagella coming out from the dinoflagellate saying, I'm bored of you, leave me alone. And it had enough space and started moving around. And we were chasing it with Jesse to try to find it. That happened in Andros after with a student when going out, collecting, sampling, and observing some very nice stuff that we spent several hours in analyzing the samples. A takeaway message that satellite monitoring is a powerful tool that is helping us to monitor the whole Earth, and the data that we get finally help with decisions in management and policy, and it can help. One of the beauties with satellite-derived information is that you can go backwards. You can go back in a region and time that it was totally boring back then. But suddenly it became very important due to another finding. So being able to revisit, it's something that you cannot do with in situ data sets. And the satellite observations, for me, I consider them as in situ data remotely sent from space. And they're constantly improving. The Earth is warming, we're responsible for that. That warming has direct and indirect impacts. Phytoplankton are invisible but vital. These organisms are essential to Earth. And the final takeaway message is that there is only one ecosystem, is called Earth. Any part that we alter and we change, eventually it echoes and resonates the trophic levels upward in different ecosystems. I would like to say a huge thank to the whole international community that we have been working all these years together. Of course, what I presented today, it's not my work, it's our work. So I'm very, very glad that we have had many fans all around the globe and a few beers, and we were enjoying with all my friends. And thank you very much for your attention and for accommodating me, for inviting me. And I'm super happy that I'm going to spend my time until Christmas for the sabbatical. And it's great to see you all. If you like me to pop in any lecture for 10 minutes, 3 hours, let me know, I'm going to do it. My program is pretty open. Thank you very, very much. 54 minutes, I'm sorry.
We have a few questions, just a few. Anyone have a question?
Me? This is awesome. We have lots of really fun stuff to talk about. But just one thing that popped in my head with kind of your light limited, nutrient limited stuff, and I never think about the oceans, but one of the issues with solar geoengineering that's being considered is not only are you changing the amount of light that's going to come down, but you're changing the quality of the light, right? You're going to have more diffuse radiation instead of direct. Does that, and I know that's a big concern with crops on the surface. Is that a concern with stuff underwater and phytoplankton? Is that something that you think would be altered?
Thank you very, very much for the question. Indeed, sometimes Homo sapiens has created an issue and is trying to solve it by pretending to be the god and trying to solve the whole thing. Usually when we play with these things, we have negative outputs. Any change in light, it is changing the amount of light that penetrates. Even in the tropical oceans, the moment you have like less light at surface, for instance, let me give you a very good example to answer to your question. Mount Pinatubo, and a huge eruption far away, it caused the biggest phytoplankton bloom ever recorded in the Red Sea. Nature paper, what happened? Anomalous cloudy conditions. Lack of light. We're in the tropical place that it shows continuous light. It blocked, not for very long, for 2-3 weeks. That blocking of sun, which was like totally gray, like the typical day in the UK, it actually, due to the changes in density, it created a huge vertical mixing, reaching reservoirs of nutrients that have never come at surface. So basically, even for 2-3 weeks, if you block the sun in a region, you can have like huge impact in the environment. Even when you block it and the light penetrates up to approximately 180 meters depth. In order to reach that, it needs like direct sunlight. There are organisms and phytoplankton communities that they live below the thermocline, the community too, in a sense. So any change you have, indeed, it's going to cause issues. In the tropical environment, you also have the tropical coral reefs. So when you create shadows, 90% of their food is coming from sunlight. The zooxanthellae. They're fantastic. And I was always wondering, how come an anthozoan, like an animal and plant together that eats, absorbs direct sunlight, but also it is eating the tree to send live small stuff. How come it relies on light by 90%? And I'm going to give you an example from my lab that I cut. Not here, not here. Very sorry.
You're seasick now.
I know, but here we are. Look at that. It's the tentacles, the polyps of corals, all these orange little dots that you see that are everywhere. Look at how many million. And this is like the only tiniest tentacle. It's full of zooxanthellae. So basically, corals photosynthesize. They have this single phytoplankton in their cells that they absorb and they transform the energy. So any kind of changes in light, it is changing the whole environment. After all, the sunlight is that. So I'm very skeptical about any bioengineering changing the light intensity within the marine environment. Thank you.